All. This is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So the discussion today is continuing on with the series of chronic inflammation. Today we are going to start looking at those mechanisms, those factors that help control, regulate, and inhibit inflammation. Of course, once there is a dysregulation of these mechanisms, that is when chronic inflammation occurs. Number one. Number two, when you have the chronic inflammation therapeutics or the drugs or whatever management interventions are, they would try to intervene using these levers that I'm going to discuss. In the immune system, there are thousands and thousands of levers. So think about it this way, that every cell, every protein, every action of the immune system can be controlled, it can be amplified, and it can be inhibited or downregulated. That means the mechanisms are going to be thousands even now. And as we progress in science, there will be more mechanisms that we will know. Clearly, today we do not have a handle on managing chronic inflammations. People suffer with chronic inflammation for a very long time. That means even when we know these mechanisms, we still do not have the right answers. So that means whatever answers we'll have here are partial. Let's start our discussion. So this camera is a new camera. This is Sony Alpha 7C with a Sony Grandmaster lens 24 to 70, I believe. I think that the lens has a little less crisp picture. Tell me, how did you feel? How did you like it? This is the picture. I think it is a little less crisp compared to Nikon and Nikon glass or lenses. All right, let's continue with our discussion. So, gifts for humanity. Here are some of the references. This is drbeen.com. This is chronic inflammation book. This is another article on the inflammation. This is cytokine storm and cytokines and chemokines and what's the difference between them. This is the toll-like receptors and what do we mean by them. A good book to buy is Molecular Microbiology, that also has immunology in it. So let's start our discussion. So regulation of the immune system, just very quickly, if I can go from inside out of this picture. Inside the picture, if you see, these two boxes represent the two arms of the immune system. The immune system has two arms, correct? One is called the innate arm. We've been talking about it for years now. The other one is called the acquired arm. Acquired arm needs to be uh, targeted towards a specific kind of antigen. It has to be taught to go react to a specific antigen. Innate arm is the first responder. It does not care for what is the antigen. It is just generally going to see something offensive in the body and it would react. Now, the very first thing, innate arm and the acquired arm, they both amplify each other. For example, when some inflammogenic substance is introduced, some antigen is introduced, some allergen is introduced, some tissue damage happens, innate arm is the first responder and it will cause the acquired arm to be activated. That is an example of immune regulation, although this regulation is called positive feedback or upregulation. Here, the immune system will become active. Then the acquired arm, once it is active, it will in turn amplify the innate arm. And this is how both of those systems would continue to give positive feedback to each other and amplify each other till they have taken care of the antigen or the pathogen or the allergen or the damaged tissue. And after that, they have to then bring each other down through a negative feedback back to normalcy or homeostasis. Chronic inflammation occurs when that controlling mechanism to bring the immune system back towards a homeostatic state, a normal state, does not happen. So, how can the chronic inflammation occur? Look, there could be excessive immune response. You wanted to make 10 cells to go fight, and we made a million cells. Or we wanted to make 10 cells and they had to be active only for, let's say, 2 hours, and now they're active for 2 years. Or we wanted to make 100 cells and we wanted them to not produce a lot of chemical substances, and they are just overproducing antibodies and chemical substances and they're just continuously triggered. There are so many dysregulations that can occur. At the end of the day, it is the breakdown of the regulatory system, especially the inhibitory system, that will result in chronic inflammation. So excessive activation and the function of the innate arm to and the acquired arm to keep amplifying and controlling each other is to also make sure to prevent attack on the self. So our immune system cells, our normal tissue cells, they all work together to make sure that our immune system does not start attacking us. And when that boundary breaks down, that is when we also get chronic inflammation or we end up in autoimmune state. The duration of the attack, the location of the attack, the intensity of the attack, all of these factors can take part in creating chronic inflammation. So if I go in here, if somebody has inflammation, that inflammation can persist if the offending environmental factors are not removed. For example, if there is mold and that is causing continuous inflammation, or if there are allergens. For example, I live in Cupertino area, and over here we get a lot of allergens during the allergy season. And I cannot just go and say, fine, either we have to move away or we have to have no allergens. That's not possible. So environmental factors are present, or the infection is present, or the person's immune system stays dysregulated. So first, environmental factors, they need to go away for immune system to calm down. Second, our tissue cells, our self. Interestingly, the self is also called antigen. For our immune system, just like for a hammer, everything is a nail. For our immune system, everything is an antigen. It is going to attack everything. However, for our self, we specially train it to be non-responsive to us or to be tolerant to us. And when that tolerance breaks down, that is also when chronic inflammation would occur. Then you would see, as we discussed the molecular mechanisms and cellular mechanisms, you would see that for every immune action, there are going to be triggers that will activate and promote and amplify those actions. And then there are going to be receptors and triggers on the same cells that would provide a negative feedback and try to control this cell's function and bring it back towards homeostasis. This is called the negative feedback. Now, the negative feedback can come from the action of the cell on itself. You may have seen those little cat videos. There is a puppet cat box on the YouTube where they turn the box on and a small cat hand comes out and turns it off, and they turn it back on and it turns it off again. Same thing here. When this cell is activated, its own actions can also cause the cell to become inactive. That is a controlling mechanism. This is called self-negative feedback. Then the cells in the surrounding can also act to either amplify this cell's function or to inhibit this cell's function. And time. There are cells in our immune systems that become activated or that are started at the same time when there is an antigen or an allergen or broken tissue. But these cells will not become active right away. They'll wait for a few days and then they will become active, and they will be regulatory cells, and they would say, fine, enough damage done for a couple of days or 3 days or 4 days, or enough damage done for this antigen. And now the antigen seems to have gone, and now we should all calm down. So imagine if there is a war, and as soon as the war starts, right away, the cells come in and say, okay, stop the war, and they hold a person, in a fight, they hold one person, the other person would keep beating them up. So what happens is our immune system actually lets the cells run for some time, take care of the offending agents, and then it tells the cells to stop. So that is the duration. And then there are many more mechanisms. For example, there is self development, tolerance to self. There is immune exhaustion as a mechanism. There are genetic factors that can suppress the immune system, or kind of make it not able to respond well. Or there are genetic factors where the immune system responds more than it should be. There are memory cells that are built all during our life. And the more memory cells we have, the more reactions we start developing. And then there is mimicry that happens. So whenever we are exposed to some pathogen, that can, not everyone, not every pathogen, but some pathogens can cause our immune system to start attacking our tissues. And that mechanism is called molecular mimicry. So these are high level, bigger mechanisms. Now let's see some of the mechanisms in detail. Tomorrow, we will do a very important part of these mechanisms, and that is T regulatory cells, the most important part of immune system. And today is going to be the grounds for understanding that. So here, before we go into these mechanisms, quick overview. We know that there is an innate arm. Innate arm of the immune system has macrophages, dendritic cells, neutrophils, natural killer cells, right? Many other kind of cells too, but these are the main players. Then we know that we have acquired arm, and the innate arm activates acquired arm. In the acquired arm, we have helper cells, we call them T helper cells, and we have B cells, and we have cytotoxic T cells, correct? Now, in the T helper cells, we know that we have a naive T cell that engages with a innate arm cell, and based on the interleukin present, for example, interleukin 4 if present and interleukin 12 if absent, then the naive T cell becomes the T helper 2. T helper 2 in turn would secrete interleukin 4, 5, and then cause the B cells to become active B cells, which are called the plasma cells. And then plasma cells would make antibodies. We know these mechanisms. That is why I'm not going in detail. On the other hand, T cell, a naive T cell, in the presence of interleukin 12, which is usually secreted by the innate arm cells, it converts into T helper 1 cell. T helper 1 cell in turn then causes T cytotoxic T cells to become active, correct? Now, today I'm introducing two more cells here. From the naive T cell, we can also end up having T helper 17 cells and T regulatory cells. We've been studying immunology together for 2 years. These two cells we have never talked about. So in the chronic inflammation and control of inflammation, we are going to talk about it. These two cells will talk tomorrow. Today, I want to make sure that we have a high level picture of these mechanisms. So now, some mechanisms, not all. Immunology is filled with thousands of mechanisms. First of all, healthy cells and immune cell interaction. We know that when healthy cells interact with immune cells, we know that there is an antigen presentation, or there is a binding of the immune cell with the healthy cell. Then what happens is there are co-stimulations that are needed, and we have done those discussions in the past. Co-stimulation will mean that we have to do a double check and a triple check. We have to stimulate in multiple ways. I call them hugs and kisses. So what happens is there are more stimulations that have to occur for the immune cell to make sure that when it becomes active, it is correctly becoming active and it is going to attack the right thing. On the other hand, if the tissue is healthy, the tissue is going to put a brake on the immune cell's activation by activating certain molecules on the immune cell to say turn off. This is like if somebody is coming to attack you and it's a robot, and the robot comes and holds you, and before it attacks you, you bring your finger out and you turn the robot off. And now you have given it an inhibitory signal and the robot is off. Now that is a normal behavior of a healthy tissue's interaction with the immune cell. On the other hand, if the same healthy tissue became unhealthy because it is now infected, let's use infection as an example. There can be many other factors, but let's say infection. It is infected. Now, when it is infected, then what happens is that as the immune cell comes in and connects with this now unhealthy tissue, the unhealthy tissue does not put brakes. It does not cause the molecules in the immune cells to become triggered to stop the immune cell. And the result is the immune cell continues to become active and inflammation would start. Now, this can become abnormal as well. This could be that the tissue is normal here, but the immune system cell, the brakes that are engaged by the normal tissue to the immune system robot to say, you stop, the immune system cell may not even have those brakes. That is a genetic problem. Or the healthy tissue cell may not have the ways to apply those brakes. That's a genetic problem, for example. And all of a sudden, they both do not want to harm each other, but they would end up harming each other. And that would cause chronic inflammation. Then, interleukin 10 is a very important factor. I'm picking and choosing those factors that are usually modulated as part of trying to control the immune system. Interleukin 10, we have done this discussion in the past as well. So let's look at this diagram once more. Interleukin 10 is released by activated innate arm cells. For example, the macrophages, natural killer cells, dendritic cells can release interleukin 10. Interleukin 10 in turn turns these cells off. This is like a robot. You turn the robot off, and the robot, when it is on, it brings its hand to a button and turns itself off. So these cells do not just turn themselves off, but they regulate themselves. So interleukin 10 is actually released by the activated innate arm cells. And that release actually causes the same cells to become less active. That is a negative feedback mechanism. That is a regulatory mechanism, inhibitory mechanism. Just a quick uh, clarification on the terms. Regulatory mechanism or regulation of the immune system means both positive feedback upwards or negative feedbacks downwards. That all of these mechanisms are called regulations. But inhibition or suppression means to bring the immune system towards a reduced state of action. So here, interleukin 10, Alexander Cotter says, did Dr. Bean listen to the Terminator movie over the weekend? No, but this is a very decent example that the robot just turns itself off. Okay, so because we don't do that to ourselves, we should not do that to ourselves. So here, the innate arm cell releases interleukin 10. That interleukin 10 causes the functions reduction. We also, the T helper cells can also release interleukin 10, and that way they can also reduce the function of the innate arm. I had discussed this, I think, a year ago in the SARS-CoV-2 context, that T helper 2 can produce interleukin 10, which in turn causes the innate arm to be reduced in its function. There are some doctors who use this kind of information to say that the acquired arm will suppress the innate arm. There is no suppression. This is modulation. Both of these arms would continue to modulate each other and balance the function. So interleukin 10 is very important. Then, and we once again will talk about regulatory cells tomorrow to know more about the interleukin 10. Then, this is also a very important function here. Imagine that this is a natural killer cell or dendritic cell or a macrophage, innate arm cell. This cell can have suppressors of cytokine signaling or SOCS, SOCS. What does that mean? This is beautiful. So stay with me for a second. It will make a lot of sense. Imagine this is a dendritic cell. This cell becomes activated. We have done this discussion, I think, the last lecture, phagocytosis. This cell becomes activated when its toll-like receptors are activated. Toll-like receptors in turn are PAMPs and DAMPs and many other kind of receptors. The function of these receptors for this cell is to identify offending foreign substances. I should not say foreign. Sometimes these cells attack our own tissue as well, which is not foreign. So just offensive agents or antigens. So once the toll-like receptors are activated, we know from our past discussions that activation of the toll-like receptor activates the macrophage, correct, or dendritic cell. And when it is activated, the macrophage of the dendritic cell, it produces a bunch of interleukins and the cytokines. It activates the acquired arm and it starts eating up other cells and it becomes active. Do you know at the same time, this is so funny, at the same time, it also starts the mechanism to turn itself off. So activation of toll-like receptor on one end causes this cell to become active. At the same time, it starts to cause the expression of a receptor called SOCS receptor, or suppressor of cytokine signaling receptor. This receptor, again, I'm going to give you the example of a robot. So imagine you turn a robot on and the robot inside of it starts making a button that it will be done making in 2 days. Then when it is done making, it will extrude that button on its surface. And now that button can be pressed to turn it off. Do you know why I love this body's mechanisms? Can you imagine how beautiful these mechanisms are? So, as the toll-like receptor becomes engaged, there is something offensive present, some antigen present, and the cell becomes active and it starts doing its fight. At the same time, now it is making these new receptors. Those receptors would start appearing on the surface of the cell in a few days. When they start becoming apparent on the surface, now what happens is that when these are engaged, they will cause the cytokine receptors to become disabled. What does that mean? Look, when this cell is active, on its surface, there are cytokine receptors. So imagine there is this mobine, I'm a macrophage, and standing next to me is another macrophage. That macrophage got activated. That macrophage released some cytokines that would act on me and activate me as well. Right? So I have to have cytokine receptors. Sometimes, as I showed you a few minutes ago, sometimes my own released cytokines will act on me to either amplify my function or to suppress my function, like we saw interleukin 10. So the cells have cytokine receptors on them. Those cytokine receptors are used to become activated. And this extra receptor that is formed, or extra protein that is formed, it will turn off the signaling of the cytokine receptor. That signaling, I mean, for those of us who are from medical field or healthcare students, the cytokine receptor signaling is JAK-STAT type signaling. So there is a protein on the inside that is Janus kinase type protein. And when the cytokine comes and attaches to that receptor, the JAK-STAT system internally becomes activated, sends out the messages, which will then translate into action. That action may be that this cell is going to go and eat other cells, or this cell is going to release some cytokines. And now this SOCS is going to turn that off. Isn't this beautiful? So this is an example of suppression of an innate arm cell by beginning of its own activity. Good. Now, this is just one example. There are hundreds and thousands of cytokines and kinases and phosphatases within the cell that help regulate its functions. So I'm just kind of bringing us to the highlights of the immune regulation. So every cell, every protein. So you would say, well, immune systems have proteins. Yes. For example, B cell produces antibodies. Antibodies are proteins. For example, innate arm, when that becomes active, when interleukins are produced, those interleukins go and act on the liver, and liver produces what we say acute phase proteins. For example, C-reactive protein. So yeah, there are proteins that take part in immune system. There are cells that take part in immune system. So all of those cells and all of these levers have regulatory actions on them. Okay, continuing. Next part is interferon gamma. And interferon gamma is going to be very popular as well for the folks who modulate immune system, for the doctors who are immunologists or who work with immune dysregulations. They would love interleukin 10s, interleukin 1, 6, chemokines, interferon gamma, interleukin 12. So here, interferon gamma. If you can keep one thing in your mind for the whole immune system, just one concept, interleukin 12 interferon gamma axis. Remember this term. I'll explain that right now what it means. When innate arm is active, the innate arm cells release interleukin 12. Interleukin 12 helps to activate the acquired arm, especially the cytotoxic site. In return, when the acquired arm becomes active, it reduces, especially again the cytotoxic site, they produce interferon gamma. Interferon gamma coming from the acquired arm in turn causes the innate arm to become activated. I love immunology and the mechanism so much that I would just like to talk about them forever. So anyways, the T helper cell 1 cells produce interferon gamma. Interferon gamma in turn goes and amplifies the function of the innate arm cells. We saw with the interleukin 10 that the acquired arm was suppressing or downregulating the function of the innate arm. Here you are seeing interferon gamma that is upregulating the function of the innate arm. And interferon gamma is not only produced by the acquired arm, to the dismay of those doctors who say acquired arm is going to somehow inhibit the innate arm. Interferon gamma is also produced by the innate arm cells itself, especially natural killer cells. And what happens when the natural killer cells produce interferon gamma, or when the T helper cells produce interferon gamma, and there are many other cells that can produce it, the result is the activity of the innate arm cells increase. What kind of activity increase? For example, interferon gamma's production causes, or action causes, more production of major histocompatibility complexes, MHC2s. MHC2 are these molecules with which an innate arm cell presents the antigen to acquired cell. So if they are presenting more antigens, the inflammatory reaction will be more. So interferon gamma is an important upregulator of the immune system. If for some reason the macrophages become dysregulated, or helper cells or NK cells or other cells that produce interferon gamma, they become dysregulated, what are we going to end up with? Sort of chronic continuous inflammation. One more mechanism. I think you would love it. This is the last mechanism for today. And again, busy diagram, but actually is very simple. And I think you would love this mechanism. Okay, so there are nice, good comments. So thank you very much. Today's the new camera. I am actually afraid that is my audio actually relaying or not? Am I just speaking to myself in the studio? But if you are hearing me, last mechanism. Tomorrow we'll talk about T regulatory cells. Today I wanted to give you a taste of what are other mechanisms. So check this one out. B cells. If I go back here for a second, remember these B cells? B cells become active to produce antibodies. That is the humoral arm or humoral part of the acquired arm. There is a cytotoxic part of the acquired arm as well. We're talking now humoral arm. And what a beauty this mechanism is. Check this out. Here is a B cell. This B cell has become active. And because of that, it looks like this little pregnant cell because it is making antibodies. Because of that, its cytoplasm is more and the nucleus is pushed to one side. This is like a little factory where things are being made very fast. And so everything is moved to a side and the place is made available for the products that are being made. Because of that, this cell looks like this. So there are antibodies. So we know that when a B cell becomes active, how does it become active? Let's start from here. An antigen, let's say SARS-CoV-2 or a streptococcus or a staphylococcus or adenovirus, something. When an antigen comes in and attaches to the immune cells, it attaches to almost all kinds of cells, including B cells. So when the antigen is present, and then B cell gets co-stimulation from the helper T cells, B cells become active, right? Helper T cells would release interleukin 4 and interleukin 5, and they would activate the B cells by proliferating them and differentiating them. For our discussion, they become active B cells. When they become active, they're called plasma cells. The plasma cells are now releasing antibodies. These antibodies are going to go and catch the antigen. That is their function, correct? So they're going to go start binding with the pathogen or whatever is the immunogen that is there, right? So they're going to start binding to it. The binding of, so see here, the number 2, the binding of the antigen with the antibody makes antigen antibody complexes. This is like you send off another robot antibody that goes and binds with the pathogen, and now they're both tumbling around. So these are the complexes, antigen antibody complexes. When these complexes are formed, we have done the discussion separately what happens, but here today is the B cell regulation with that. So check this out. Arrive on this side now. So now we have B cell has released the antibodies. Those antibodies have connected with the pathogen. We have complexes. Those complexes will actually connect back with the same B cell. How? Remember in the mast cell discussion, we discussed that the cell, mast cell, has a receptor on it that can bind with the fraction constant of the antibody. So we know that this is an antibody, right? This antibody has a variable region where it binds with the antigen. And it has a constant region where biological actions are taken. Antibody can bind with various cells at its FC part, fraction constant. This is fraction variable because it binds to various kind of things. But this fraction constant is constant. It's the same for all antibodies. What happens is, by all antibodies, if it is IgG, then all IgGs have the similar, there are some variations. IgE may have different, IgA may have different, but all IgGs of one class would have the same fraction constant. That FC part then binds to the FC receptors to attach to the cell surfaces, for example, mast cells. They also attach with platelets. And here, the fraction constant, imagine this is the complex, antigen antibody complex. The fraction constant connects on the B cell with the FC receptor. So this receptor is called, check this out with me, I'm going to make it bigger. It's called FC, fraction constant, gamma for IgG, G for gamma, receptor R, receptor type 2 on the B cell. So FC gamma R2B. That is a receptor here on the B cell surface. FC part of the antibody will attach there. Now, What is this antibody holding? The antibody is holding the pathogen or whatever is the immunogen, whatever is the offending thing. That offending thing, remember, was also connecting with the antigen receptors, which in case of B cells, antigen receptors are actually IgM and IgD. But forget about that for a second. Antigen receptors. So here, the antibody is holding the antigen, and that antigen cross binds with another antigen receptor on the B cell, like a short circuit. So the tail, so there is this complex, right? The antibody tail, I'm calling it tail, FC part, is binding with this receptor, while the antigen that it is holding is binding with another receptor. The result of this kind of binding on an active B cell is that there is a signaling pathway inside the B cell that is started. That is called ITIM. It's an inhibitory molecule within the B cell. Once that becomes activated as a result of this binding, and you know what it happened, what it does? That activated molecule inside the B cell goes and blocks the antigen binding cell receptor signals. What does that mean? That means it tells this B cell, even when there are antigens on the outside, it tells the B cell not to respond to those signals and do not become more overly excited, as I am today with this lecture. So, will the B cell continue to work? Yes. Will the B cell make more antibodies? Will become more excited? No, because there is this regulatory mechanism that is going to bring the B cell down. Now, think about it. If somebody's B cell receptor for Fc gamma isn't working correctly, or somebody's Fc gamma does not bind here correctly because there is a problem, genetic problem for producing the correct IgGs, then will this B cell stop working, stop making antibodies? No. I'm going to say, fine, I have become activated. I'm now going to continue to produce antibodies. And continuous production of antibodies is going to mean chronic inflammation. Fortunately, this is not very common, so we are okay, but this is the B cell regulation. So with all of these, this was the last point I wanted to make. Tomorrow, we'll look more on the T cell side, and we'll look at the T regulatory cells. Today, we saw interleukin 10, we saw interferon gamma, we saw B cell activation or inability to activate. We saw the macrophages and dendritic cells in the immune system cells and how do they activate or deactivate each other. So with this, I will see you on the chit chat in a few minutes on the Cool Beans Cafe live channel. That's a different channel where we just huddle together and do a little chit chat. So we'll see each other over there. Please do me a favor, like, subscribe, and share. There are links in the description. One of them is to buy access to Dr. Bean at a very, very affordable price. It's lesser than $100. Although I was talking with my team that this is time to stop that sale because that is too low of a price for 1,000 lectures. I think every one lecture is worth more than that. So anyways, take advantage of that. There are other links in the description as well if you would like to buy me a coffee or if you would like to use PayPal to support this work or become Patreon or become a Substack member. Thank you very much, and I would see you on the Cool Beans Cafe live site in a few minutes.